welcome to another exciting episode of Novelty. Today we're here at the National Commission on Research, Science and Technology, and we're here to learn about biotechnology. We're even clearing up some misconceptions, talking all about biosafety and basically everything that falls under biotechnology. My name is Diana Master, so follow me as we go learn a little more about biotechnology.
Good morning everyone, good morning Diana. I am Gwyneth Farmer and I am currently an intern at the NCRST within the Biotechnology division.
All right, so since we are talking about biotechnology, what is biotechnology in layman's terms, please?
In layman's terms. So just to put it simply, biotechnology is a technology that uses living organisms and parts of living organisms to develop new products. So a simple example of biotechnology can be baking or brewing, because with baking we make use of the living organism yeast to produce bread. Mm-hmm. So quite simple.
Very
actually.
Simple.
Yes.
So why did you choose a career in biotechnology?
I chose a career in biotechnology because it's actually a new innovative field within the science field, and there's so much that can be used from biotechnology within different industries. Mm-hmm. Not just food science, but also within the medical field. Biotechnology is very important.
Awesome. If you had to choose one organism to study for the rest of your life, what would it be and why?
Okay, such a great question, by the way. So I am currently doing my master's at the University of Namibia and I am studying viruses. So what I would love to study for the rest of my life basically is just viruses. They're quite unique because what people don't know about viruses is that on their own they're not living organisms. They require a host organism. So it's just very fascinating how they are able to cause such big diseases such as COVID, for example. So
and with the mutations, that's also
and how they adapt, and how we've been struggling with HIV, which is also a virus, since the early 80s. And it's still an ongoing problem. So that's why I find viruses so interesting. Because I think, like you're saying, viruses always are constantly evolving.
They're constantly evolving. Yes. And now obviously because of global warming and all of that, we're unlocking basically new organisms, which means new viruses. Terrifying.
It's very terrifying, yes.
So what are the 5 benefits of biotechnology?
Okay, so from what I've gathered by my research so far is that biotechnology can, as I mentioned previously, can be used in different industries. So one of the industries that I'm focusing on is medical advancements, because you can use biotechnology to produce regenerative organs. It can also be used with climate change, combating climate change, by converting biofuels from wastes within the ocean. And then lastly, another benefit of biotechnology, it can also help with food production with the use of GMOs. And since I am in the GMO division at the NCRST, we focus on the production of GMOs using plants, and this can just help to aid with food production, like I mentioned, and also just with the development of faster growing crops, and crops that have higher nutritional value.
I like that you brought up GMOs.
Yes.
Because there are a lot of misconceptions about GMOs. For example, people think that GMOs are what makes them fat.
No.
They believe that GMOs are detrimental to their health, but I guess that's where the NCRST comes in, so that you flag these things.
Yes.
Okay.
So at the NCRST we do have a biosafety department, and they are responsible for basically testing these GMOs and bringing those myths, basically just killing those myths.
Yeah, okay.
Because your body cannot be modified by a GMO from eating them, for example.
Okay, I see.
That's
you see, it's a misconception. People believe that that's what GMOs do. I mean, we've been hearing that GMOs are bad for us and all of that. So, it is something that you would need to clear up.
Yes. So a GMO basically, all that can maybe change with a modified plant for maize maize, for example, what they can do is they can just insert a gene that makes this plant an insecticide, basically. So that doesn't mean if you eat the plant, you are now an insecticide. No. It just means that it aids the plant with combating different insects that try to eat it.
I see. Okay.
So that's just it.
All right. Yes. Okay. Now, before I let you go, what are your future aspirations?
So as I mentioned, I am doing my master's, and I would really love to continue studying. I do see myself in 2 to 3 years' time at UCT because they have a very great viral study department, and they focus a lot on plant development, which is what I am in now currently. So I would like to continue with my PhD in 2 to 3 years' time.
And soon you'll be doctor.
Soon I'll be Dr. Farmer.
Dr. Farmer, we'll put some respect on your name.
Thank you so much.
No, thank you so much for giving us a little insight on biotechnology and what it is.
No, you're so welcome. And thank you everyone for watching my segment.
All right, stick around. More interesting stuff coming up.
Good morning, everyone. My name is Dorothea Shailemo. I'm a senior scientist in the National GMO Testing Laboratory under the Biotechnology Division here at the NCRST. So I just want to give you a background of why this lab was established. The purpose is really to assist with the implementation of the Namibian Biosafety Act, which is Act 7 of 2006, which provide for measures to regulate genetically modified organisms and their products within the country. So the Act requires that we monitor genetically modified contents in our food and feed products so that we don't have genetically modified organisms and products that are not authorized for use in the market. And also just for consumer choice, so that the consumers have a choice to whether they want to consume genetically modified organisms and their products in the food. All right, so basically what the lab does is perform GMO testing on food and feed samples just to determine the content of GMOs in those products and whether they comply with the regulation requirements. Because as per the Act, it says that genetically modified products and those products that are above 0.9% should be labeled for consumer choice. So the lab also does verification. So our inspectors go to the fields, to the borders, to millers, pick up samples, and we test them for GMOs. So that is the reason why the lab was established. So I'm just going to take you through the process. First, for you to test for GMOs, you need a sample. So you start with a sample. A sample can be a seed, a feed sample that you need to extract a genetic material from that sample that you use for PCR analysis, which is polymerase chain reaction. It's a molecular analysis which allows you to determine whether that sample contains GMO or not. So DNA is like an input for PCR. And the reason why we are here today is for me to demonstrate how we go about DNA isolation from food samples or feed samples. We start off with seed samples. As you can see here, these are maize seed samples, but they are pre-coated just for preserving them and to prevent them from being attacked by pests and other that may. So first, for us to get to DNA extraction, we need to prepare our samples because DNA extraction requires a uniform kind of texture in order to access the DNA, because if you start with the seeds, it won't, the DNA extraction will not be successful. So you have to prepare the seeds, which means you have to crush them into powder, like fine powder, and we do this just using a normal blender, like it's the same as the one that we use at home, but this one is industry grade. Okay, so you put your seeds in the blender, close it properly, and then you just after your extraction, there is these discrepancies between these two, then you know that something was wrong. And we also perform our extraction with extraction control for us to be able to trace back where we contaminated our samples, for example. So yeah, so for extraction, we use a gene spin isolation kit from Eurofins. So the kit comes with all the reagents that you need, and it also has spin columns, so that at the end of your during the process of your extraction, you have your DNA binds to this membrane inside the column here, and then after washing, the DNA is eluted into a new column, and that DNA is ready for subsequent reactions like PCR. This equipment, as tiny as it looks, it's very important to us because it helps us determine how pure our extracted DNA is, and the concentration also, so that we know whether we have to repeat this extraction or not. So what we do first with this spectrophotometer, we measure the blank, and usually with blank we just use our elution buffer, because that is the solution that we use to dissolve our DNA from the membrane in the spin column. So now I'm just going to perform a blank first before I measure. I'm not going to measure, sorry, before I measure the samples. So, and I'm going to be using this elution buffer. So you pipette 5 microliters, and then you close the arm, and then you just click measure. It's already on blank, and then it gives you the readings. That is now the concentration of your blank and also the ratio. So like I said, the acceptable purity range of DNA is ranges between 1.8 to 2, and if it's lower than that, it indicates that there are other contaminants. If it's lower or higher, it indicates that there are other contaminants in the solution, and you should either purify it further or you repeat the whole process again. So now I am done with blank. I'm just going to measure now the first sample, just for you to see how it looks like. This is how it looks like. So now you have your concentration, the concentration of your DNA. In this case, it's 149.2 nanograms per microliters, and the ratio is 1.76, which is like slightly close to 1.8, so it's fine. So that means you can use this DNA for further analysis, this PCR. So yes, this concludes the whole DNA extraction process. From here, you can choose to do either gel electrophoresis if you want to see the bands, see how much of DNA is present in your sample. But in this case, what we do is we proceed to the PCR room, where we do our PCR analysis detection now for genetically modified organisms in our samples. And we use Eurofin kits. It comes with a plate that you just aliquot your samples into the plate. You already have reagents within those holes, and then you insert the plate into the machine. Then the whole process takes around 3 hours, and then after 3 hours, it shows you this. It shows you the readings, but you are also able to see the process, the reactions on the screen. So it's like a real-time PCR machine or a real-time process. You see how the amplification is going on. So the PCR, this DNA is now amplified into millions of copies of your gene of interest. This allows you now to detect GMO genes in your DNA, or GMO DNA in your sample. So yes, this is the end of the DNA extraction for GMO analysis.
My name is Panduleni Ngitila. I'm a biosafety inspector at the National Commission on Research, Science and Technology under the Biosafety and Biotechnology Division. Today I'll be giving a brief rundown of the emerging technologies that are currently happening within the biotechnology segment. Biotechnology is really a diverse field, so I'll try to be brief as possible, and also these technologies mostly they interact with each other, and they both stem from common ancestors, if I may put it like that. So to start with one, I'll go with artificial intelligence. I know artificial intelligence is quite widespread and it affects a lot of areas. That includes biotech as well. So within biotech, AI is used in diagnostic medicine to sort of diagnose patients in automated processes. And furthermore, it also helps on the research side when you are trying to discover new medicine or to delve into the effectiveness of therapies. AI sort of simplifies the work of scientists by amalgamating the data and just extracting the relevant portions that you need, which gives you the exact and direct information that you need. Furthermore, biotechnology is actually very critical in artificial intelligence because it enables machine learning. Right now, AI in other fields is being used sort of for analysis, especially in the accounting sector, but it's sort of limited to that because it lacks that human part of emotions to make decisions that require cognitive dissonance to say where you need to feel and decide. However, if you infuse biotechnology into AI and you understand the biochemical mechanisms, the emotional substrates that lead up to making a decision, then we stand to make AI more informative and more responsive, as well as applicable to a wide range of industry, including law, etc. And then to move on to the next, I would go with now gene editing, which of course includes the CRISPR or Cas9 cassette, which is really a way of getting into the genome of an organism. Genome, by the way, is the entire genetic makeup of an organism. So this technology allows you to get into the organism's genome, delete or insert a certain type of gene or DNA, which then allows you to create an organism with enhanced characteristics that differs substantially or not substantially from the original form. So it enables you to give new functions to an organism. An example I can make is GMOs, genetically modified organisms, where, for instance, you have your maize crop, and then you pick up that your maize are being affected by a certain type of crop, or pest, or you figure that they are not resistant to the glyphosate, the herbicide is probably killing them as well. So what the scientists do, they go to the lab, they take a bacterium which is resistant to that pest, and then they insert it into the maize crop, and then it's then expressed into the maize crop. Then when you go out there and plant your maize crop, it's now resistant to these pests. To go on to the next one, it is now gene sequencing. Again, like I said initially, these technologies, they sort of learn from each other, they utilize each other in one way or another. So with gene sequencing, this one allows you to map out the whole sequence of any organism, be it a virus, bacteria, a human, because currently as we speak, the human genome has already been mapped out and people know exactly which genes human beings contain, which ones are responsible for what, and so forth. So with gene sequencing, remember when we had the novel coronavirus, I'm sure most of us were keeping up to date to say there's now a different mutant, it's now COVID variant 2. It's because of gene sequencing that people were able to tell that this sequence differs significantly from the previous virus, which then enables you to tell that a mutation has occurred and this is how to best deal with it. And then again with gene sequencing, it also enables you to study and understand in depth to say the function of each gene in the genome, because once you map out the whole entire genome, you can then remove one gene and then you observe the organismic function to say that because I removed this gene, this organism is now not able to metabolize lactose, for instance. Then you know the function of that gene is to metabolize lactose. That's basically about gene sequencing in a nutshell. Now, moving on to industrial processes, there is what we call biomanufacturing, which is really using living organisms to produce stuff that they would not be able to produce on their own. Again, like I say, they borrow from each other. This one will sort of utilize gene editing. So with biomanufacturing, an example I can give is insulin, because this is mostly applied in the medical sector. So we all know insulin. There's a type of diabetes. There are two types of diabetes. The one is where your body cannot produce insulin. The second one is where your body produces insulin, but it's not being picked up by your beta cells. So in the first case, it can be remedied by injecting yourself with insulin. So what scientists have done, they have isolated the responsible gene for creating insulin from the human body, and they've inserted this into a bacterium. Now you have a bacteria that is capable of producing insulin. Once this insulin is produced, it is then extracted and purified, and then it's sold on a commercial basis.
Today, my name is Tristus Balakia, and I'm studying a major in Bachelor of Communication at NUST, and I have a passion for ICT.
Good day everyone, my name is Miriam Shikongo, a Bachelor's of Arts in English student at the University of Namibia, and my interests are digital marketing. Well, I decided to enroll or be part of the Seeds for Future program because it indicated that they were going to inform us on topics such as artificial intelligence, cloud and digital power, of which my interest lies in digital power.
So I was actually selected for the program, but I obviously couldn't turn down the opportunity because it gives you the ability to deepen my understanding in the ICT industry and connect with like-minded peers who are eager in the city as well, and the opportunity to learn from industry experts.
My love for ICT stemmed from my interest of digital marketing. I would say I've always wanted to learn how to do graphic designing using various softwares, and I think that's where my love for ICT stemmed from.
My passion for ICT stems from its transformative potential, because it's an ever-growing industry and it will keep on growing as we go. As for now, we are looking towards AI and all that, so it keep on growing as we go on, and it's actually an advantage to gain this knowledge and understanding, because you will need it in everyday life, basically, workplace, personal life, everywhere. And yeah, basically that's it. What I'm looking forward to the most is just to connect with like-minded peers, like I said before, and to gain an insight on Huawei's technological advancements, and to learn from industry experts, and yeah, just to broaden my knowledge and understanding and gain first-hand experience in the ICT industry.
So as I've mentioned, my interests are in digital marketing, and so I'm looking forward to digital power so that I can learn from experts, the Huawei experts, as I believe they have a high experience in with regards to digital power, and that's what I'm looking forward to.
The Seeds for the Future program is Huawei's biggest global flagship corporate social responsibility program, which aims to develop the local ICT talents, enhance knowledge transfer, and promote a better understanding of telecommunication sector among the youth.
Well, what an exciting, insightful episode we had today. We learned all about biotechnology, even managed to clear up some misconceptions. But that brings us to the end of our episode today. Thank you so much for watching Novelty. You can join us again next week, same time, same place.